So this is going to have quite a bit of biology to start with. So I work with some collaborators in Manchester who work on Drosophila, and they work on axons and neurodegeneration. So your brain, your nervous system is wired by neurons which have very thin projections called axons that come out of them, and they're up to 1 meter long but only 1 micron in diameter. And they contain bundles of a protein called microtubules, or a structure called microtubules, that act as a structural backbone and transport highways for transporting material down the cell. And what happens, what they see during aging or neurodegeneration is that you can see local swellings where instead of the microtubules being nice and straight in these bundles, you see local regions where the microtubules are all curly and twisted around. And that is extremely bad in terms of the function of the cell, because the microtubules are used to transport material from the nucleus down to the axon terminal, as well as back again. So what are microtubules? So microtubules are formed from dimers and then protofilaments and then microtubules, so a helix of a protein called tubulin, and they're very, very dynamic. So this is some microtubules grown in vitro, and so their average half-life in vitro is about 5 minutes. And they're regulated by a lot of different proteins. It's about 300 odd, more than that, different proteins, which are involved in basically every different aspect of microtubule in all of your different cells. And it's the deficiencies in a lot of these proteins are known to induce neurological disorders. So these are the kind of regions that they see. So if you take wild type flies, they look sort of nice and linear. You knock out lots of different types of genes that are involved in microtubule regulation, and you see after the microtubules have initially been laid down nice and straight, you end up with these regions where the microtubules are suddenly going all chaotic. So this is bad. And so these are very similar in lots of different cases. The kind of they look very similar in lots of different treatments that they can do. Right, so what do we know about microtubules? Microtubules have this high persistence length. They are the stiffest of the cytoskeletal polymers. And so their persistence length, the length scale on which you expect them to act like a rigid rod, is of 2 to 6 millimetres. So when you look at them in gliding assays, which is a typical in vitro assay, so you have a protein called kinesin, which is a motor protein that normally it carries cargo down microtubules. In these motility assays, you bind the kinesin to the surface and then let them move the microtubules around. So what that looks like is normally the microtubules just glide in a straight line. So they are stiff, their persistence length is such that you basically see them going straight. However, in vivo we see them with these really highly curved structures. And in order to sort of bend the rigid rod into such a tight curl, that would need a lot of energy if they're just rigid rods. But you also sometimes see them in the motility assays. So you can see, so this is a motility assay in crowded conditions where there are lots of unlabeled microtubules. There's lots of extra forces being acted on the microtubules. And you see a microtubule come in from the top, and then it bends goes into a circle, goes around in a circle for a while, and then manages to unravel and goes on straight. Whereas the one next to it just carries on in the circle the whole time. And this sort of stills of this kind of thing. They're not pinned to the surface. They can move around within the context of the this looping behavior, and their loop radius is sort of 1.5 micron, the same kind of order. So I'm trying to model like that, and I think that this behavior could be regulated by the microtubule associated proteins, and in particular, this assay is done with kinesin. So several associated proteins have been shown to be sensitive to the curvature of the microtubule, so that they bind more to the curved parts. So you can see, like here, there's an increase in the amount of bound tau in that case. And it's been shown for kinesin to have that effect. Recently, there was an experiment from Warwick where they take a microtubule. So this is a normal microtubule attached to a cover slip, and they put a flow. So the flow bends the microtubule, and they also add an inactive version of kinesin. So this is the same kind of kinesin that's used on the assays, but it doesn't walk along the microtubule. And what happens when they let go of the flow is that instead of retracting, going back to straight, it only partially does. And you end up with an induced curvature from the binding. Their assumption is that their hypothesis is that this is coming from differential binding. So more is bound to one side than the other in this case. If they increase the concentration a lot, they lose the effect, presumably because it's saturated both sides. So when they have more, so the assumption is if you've got more kinesin bound to the convex or concave side than the concave side, possibly the other way around.
Is this reversible? That if you decrease the ATP concentration, for example, that...
So this is kinesin that is non-motile, so it's bound in a strong state. It is reversible in the sense of, I think it's reversible if they get it to fall off, but I'm not sure.
Okay.
Certainly, it sort of very slowly unbinds, like over long time, much longer time scales, the kind of unbinding rates. So I showed a picture earlier on that had a microtubule as like loads of these little balls, but actually those balls are these really complicated protein structures, and it's entirely plausible that when you bend the whole structure of the microtubule, you change the contacts between the kinesin, so that hence you can get more kinesin bound to the, it can prefer one side to the other. Right, so that's the biology. So I have a model for that. So I have the microtubule as an inextensible rod, and the energy depends on the curvature. So it's inextensible, so I'm not worried about it stretching as this happens. And then I use the sort of cheapest, easiest approximation I can, resistive force theory, in order to get some equations for my mechanics. So I have sort of an excess curvature. So capital K is the difference between the current curvature and the reference curvature. And then I need to specify how the reference curvature evolves. So you can see my brilliant diagram skills. So I have two sides to my two-dimensional microtubule, a plus side and a minus side, and I sort of do fairly standard binding to to the sides. So I have the kinesin on the surface. It can see both sides, and it can bind, associate, and dissociate, and a little bit of diffusion. So there's a measured diffusion coefficient, which is very low. And then this will move as the microtubule moves, because it's a gliding assay. And then to that kind of standard behavior, I put a function here so that the kinesin prefers to bind to one side of the curved microtubule. So having done that, I then need to couple back to the mechanics. So I then allow the reference curvature to depend on the difference of the binding. And then I have a load of parameters, like sort of scaling factors, unknowns. And that lets me maintain the curvature once the forces are removed.
I'm sorry, you probably said it, but on the previous slide, you are allowing the proteins to move, it looks like, with some speed v.
Yes.
Okay.
So the proteins are not moving. The proteins are attached to the surface. The microtubule will move. So that v is the velocity of the microtubule.
Okay.
Because
this is like a gliding.
It is a gliding assay. So this is the material derivative to incorporate that. So that lets me, now the microtubule, so this is sort of reproducing the experiments in Warwick where it bends in a flow, and then when you let go of the flow, it stays bent to some amount depending on how much is bound, which will depend on how long it's been going and the other parameters. So I can look for steady states. So I have sort of a system of equations. I can look for steady states, and I can find steady states where there's uniform curvature, so constant curvature along the rod, and then kappa 0 equals kappa naught equals 0 is just a straight microtubule, whereas roots of this that aren't 0 will be curved states, arcs and rings. This is an odd function here. So this is the transcendental equation I get for my steady states. So it's a nested tanh, and it behaves like kappa naught at large values. So if the derivative at 0 is negative, then an arc, an additional root must exist. And as we change the parameters, you can see additional roots appear. So initially, you just get one set of roots as I'm changing the attachment rate, effective attachment rate, which has lost the gamma from earlier. So you just get 2 points appear, but then as you increase it for some sets of parameter values, you can see additional steady states appear. So you, at these points, you can have more than one size of potential arc solution. And I can do a linear stability analysis, and I'm going to plug the compound matrix method. If you're doing sort of an eigenvalue problem with boundary values on either side, please talk to me if you're interested. It's very efficient at finding those eigenvalues without having to do numerical things, sort of discretization. And then I can plot the size of the solutions as a function of one of the like the attachment rate. So as you change some of the parameters, you see some, like, as you change the parameters, you go from just having one solution where, like right at the start, that's not quite, one solution where the steady state, the uniform solution is, or the non-zero, the zero solution is unstable, to being stable And then at these points here, you have a stable straight configuration and a stable curved configuration at the same time. And then, so I get that using numerical simulations on my PDEs. Oh, that jumped. So, I don't know quite why it's jumping. Is it going to loop? So I need to induce some initial curvature in order to see anything. If I don't, I will just get like the straight solution. And so I initially pin it temporarily and then unpin it at some point. And so what you see is it builds up a bit of curvature and then I unpin it and then it can go into this other state. And so here is sort of that effect, but only changing one parameter. Let's show that in the browser. Oh, sorry. We're just going to get to... For some reason, when I import this into good old PowerPoint, it makes it black and white. So it's exactly the same file. So this is just changing a slight tweak to one of the parameters to how long I hold it pinned for, and this one will straighten up again and this one doesn't. And then this one will start just going around, will stay in locked in this curl. And then just as something strange, I can also find some parameter values which are probably non-physical, where waves sort of propagate along the microtubule. So the talk earlier kind of reminded me of this. So what's going on with the speed of that? So yeah, you can get waves propagating down as sort of a balance between the velocity of in the material derivative and then the sort of propagation of the fact that as it curves, more of it binds.
Why do you get straight regions?
What do you mean?
Is that curvature on the...
Oh, sorry, on the right, that's the sort of preferred curvature.
Ah, okay, sorry.
So, yeah, sorry, I didn't explain that. So that's what it's trying to get to. So when it saturates with like more bound to one side, it goes to these lines. Okay, and so in summary, I see highly curved microtubules, and I'm very interested in them because they're very interested in sort of neurodegeneration context. You can see them in the gliding assays where kinesin is used. I know that kinesin can lock in curvature, and it's also the same kinesin that's used in the gliding assays, so I think it could be that that's making the loops in the gliding assays. And I have a reasonable amount of parameter space where stable straight microtubules and rings can coexist, and therefore they could switch between them, as seen in the experiments. This model fits there. Good. And then my next step is to move back into in vivo, like into the axon rather than just modeling in vitro. Now I have a sort of model for induced curvature where you can have other proteins competing with kinesin and having a protective effect and that sort of thing. I don't think it's kinesin in the axon that's doing this. I think it would be something else. But the fact that I know that kinesin can have this effect. Okay, and my collaborators, and thank you for the Leverhulme Trust because they give me money.
Quick question or two? Yeah.
So qualitatively it looks very nice. Does it reproduce for reasonable, you have 1 million parameters, So there's always a risk there,
Yeah.
as
So
we know. But for reasonable values of parameter, does it reproduce the curvature, the radius?
like the curvature is kind of a parameter that I have to tweak, unfortunately. So,
Okay.
like, I can constrain that by experiment, but I can't.
So can this model make any contact where you can compare with the experiment?
If I've made some predictions, and I'm doing some experiments. So I can make predictions as to changing this should have this effect. Like,
Okay. Thanks again.
yeah.